Chapter 16. Climate, Race, Brain Size, and Intelligence. Evolution of the races. Cognitive demands in northern latitudes. Race differences in brain size. Race differences in winter temperatures, brain size, and IQ. Brain size and intelligence in humans. QNCNWN. Contribution of race differences in brain size to differences in intelligence. 7. Sex differences in intelligence and brain size. 8. Genetical processes in the evolution of race differences in IQ. DS chapter gives an account of the general principles of the evolution of race differences in intelligence. The crucial selection pressure responsible for the evolution of race differences in intelligence is identified as the temperate and cold environments of the Northern Hemisphere, imposing greater cognitive demands for survival and acting as selection pressures for greater intelligence. The South Asians and North Africans, the Europeans, the East Asians, Arctic peoples, and Native Americans adapted to these cognitive demands by evolving greater brain size and intelligence. The genetical processes consisted of increases in the frequencies of the high intelligence alleles and of mutations for higher intelligence. 1. Evolution of the races. The consensus theory of the evolution of the races is that humans evolved from apes in sub-Saharan Africa during the last 4 million years or so. During this time, a succession of species known collectively as the hominids evolved with increasingly large brains. These were the Australopithecines, followed by Homo habilis and then by Homo erectus, who appeared about 1.5 million years ago, and finally by Homo sapiens, who appeared around 150,000 years ago. From around 100,000 years ago, groups of Homo sapiens began to migrate from equatorial Africa into other regions of the world, and by around 30,000 years ago, they had colonized most of the globe. In the early part of this period, they spread through most of sub-Saharan Africa, and by 100,000 years they were established in the south of Africa, where they evolved into the Bushmen. By 88,000 years ago they were settled in Southwest Asia. By 60 to 40,000 years ago they were established throughout Asia, and by about 40,000 years ago they were settled in Europe, the Indonesian archipelago, Australia, and the Americas. During the last 6,000 years or so they colonized the Pacific Islands. A map showing the approximate times and directions of the migrations of modern humans indicated from the archaeological record is given in Figure 1. It is a general principle of evolutionary biology that when populations are isolated from one another in different locations, they inevitably develop genetic differences and evolve into different breeds, or, in the case of humans, races. These differences evolve through the processes of founder effects, genetic drift, mutation, and adaptation to different environments. The founder effect occurs when a small group breaks away from a population, migrates to a new location, and establishes a new population. The migrating group is likely to differ genetically by chance from the group it has left, bringing about two groups with different genetic characteristics. It is not considered likely that this process played any significant part in the development of genetic differences in intelligence between the races. The second process through which races diverge genetically is through genetic drift. This is a process in which the frequencies of some genes increase, while those of others decrease through chance. It is possible that the racial differences in the frequencies of different blood groups and of genetic diseases may have arisen in this way, but again, this process is not considered likely to have played any significant part in the development of race differences in intelligence. It is believed that it is through the two remaining processes of adaptation to different environments and genetic mutations occurring in some races, but not in others, that race differences in intelligence have come about. Many of the human race differences can be understood as adaptations to climate. The morphological differences have evolved in accordance with Allen's law, which states that species and breeds in cold regions tend to evolve shorter limbs because these produce a smaller ratio of surface to body volume, and this reduces heat loss. Hence, fast Asians and Europeans in temperate and cold environments have shorter limbs than Africans in tropical and subtropical environments. The dark skin of the Africans and Australian Aborigines living in tropical and subtropical environments gives protection against sunburn and skin cancer. The absence of facial hair in East Asian men prevents frostbite that would develop if the hair froze on the face. the smaller nostrils of East Asians and Europeans, as compared with Africans and Australian Aborigines, warm and humidify inhaled air. 2. Cognitive demands in northern latitudes. The selection pressure for enhanced intelligence acting on the peoples who migrated from tropical and subtropical equatorial Africa into North Africa, Asia, Europe, and America was the problem of survival during the winter and spring in temperate and cold climates. This was a new and more cognitively demanding environment because of the need to hunt large animals for food and to keep warm, which required the building of shelters and making fires and clothing. In addition, Miller has proposed that in temperate and cold climates, females became dependent on males for provisioning them with food because they were unable to hunt, whereas in the tropics, women were able to gather plant foods for themselves. For this, women would have required higher intelligence to select as mates the men who would provision them. For all these reasons, temperate and cold climates would have exerted selection pressure for higher intelligence. The colder the winters, the stronger the selection pressure would have been, and the higher the intelligence that evolved. This explains the broad association between latitude, or, more precisely, the coldness of winter temperatures and the intelligence of the races. The theory that race differences in intelligence evolved because the peoples who migrated out of Africa into the temperate and cold climates of Asia and Europe entered a more cognitively demanding niche that required greater intelligence is a further instance of the general principle that it operated in the evolution of greater intelligence in mammals when they colonized the nocturnal niche, birds when they colonized the air, monkeys and apes when they became cooperating social animals, and hominids when they adapted to the open savanna. The new niche of the temperate and cold environments colonized by the races that migrated out of Africa demanded an adaptation from an herbivorous to a largely carnivorous lifestyle. The primates from whom humans evolved had lived for a period of approximately 60 million years as herbivores in the tropical and subtropical environment of equatorial Africa, in which plant foods are available throughout the year. The hominids that evolved in equatorial East Africa remained largely herbivorous. In contemporary times, hunter-gatherer peoples in tropical and subtropical latitudes continue to subsist largely on plant foods, of which numerous species are available throughout the year. Because primates are adapted as herbivores in tropical and subtropical environments, they have found it difficult to survive in temperate environments, in which plant foods are not available for a number of months in the winter and spring. An early instance of primates encountering the problem of survival during the winter and spring in temperate environments occurred during the mid-Miocene between 16 and 14 million years ago. This was a warm period in which much of Eurasia became subtropical. Two species of apes, Pliopithecus and Dryopithecus, migrated from Africa into Eurasia and flourished there. At the end of this period, about 14 million years ago, Eurasia became colder and the climate became temperate. In Europe and in most of Asia, these apes were unable to survive during the winters and became extinct. The only part of Asia where these early apes were able to survive was in the tropical southeast and the Indonesian archipelago, where they evolved into the orangutans and gibbons. 3. Race differences in brain size. The races that migrated into the temperate and cold environments of North Africa, Asia, Europe, and the Americas evolved greater intelligence to survive in these more cognitively demanding climates. To accommodate this enhanced intelligence, they evolved larger brains, just as had occurred in previous adaptations in the evolution of mammals, birds, and primates to more cognitively demanding niches. Studies on race differences in brain size have been given for each race in chapters 3 through 12. It is not possible to average these to give mean brain sizes because there are different methods for measuring brain size, and these give different results. The principal methods are by measuring the length, breadth, and height of the head of living individuals and calculating the volumes, and by filling skulls with lead shot or seed and transferring these to a container to measure the volume. What is needed is a large collection of brain sizes measured by the same method and one that includes all the races. Only one such data set is available. This is the mean brain sizes of 87 populations worldwide, based on measurements of approximately 20,000 crania, published by Smith and Beals. These are categorized in Table 16.1 into the 10 races with which we are concerned. The figures in bold are the means of the brain sizes of the samples of each race. Table 16.1. Brain sizes for 10 races. Race or brain size or race or brain size or race. Meta-Americans 1,366, or peoples 1,443, or Africans, or brain size, or OAM 1,282. Araucanians, Carib, or 1315, or Koryak, or M. Mises. Cheyenne 1,399, or Ostiak, 1,416 EE, or Islanders 1,317 N. Choctaw, or 1292, or N88, or New Britain, or 1232, or New 1,225, or Caledonia 1,311, 1,228, or New Ireland, or 1250, 1,250, 1263, or Ntfen 1232, 1,300, and 81,338, 338, or Ka, or QL, or AMs, or Haida, or 1358, or We, or 1404, or South Asians, or Dersim Czechs. Brain size and winter temperature, greater intelligence in the races that occupied the colder environments, is shown in Table 16.2.
Column 2 gives the races ranked by the severity of the winter temperatures to which they were exposed. Column 3 gives present-day coldest winter monthly temperatures taken from the Encyclopedia Britannica World Atlas and are averages of the regions inhabited by the races. Column 3 gives the coldest winter monthly temperatures during the main worm glaciation, which lasted between approximately 28,000 and 10,000 years ago, and during which winter temperatures fell by about 5 degrees centigrade in the northern hemisphere but not in the southern hemisphere. Column 4 gives average brain sizes taken from Table 16.1. It is apparent that there is a general correspondence between coldest winter monthly temperatures and brain sizes. For the first 6 races listed, brain sizes decrease with less severely cold winter monthly temperatures. However, in the remaining 4 races, this linear trend becomes irregular. The Africans inhabit a warmer zone than the Bushmen but have larger brain size. The Australian Aborigines continue the trend with a warmer zone and lower brain size. However, the Southeast Asians and the Pacific Islanders in tropical and subtropical zones have larger brain sizes than the South Asians and North Africans, the Bushmen, the Africans, and the Australian Aborigines. Column 5 gives the IQs of the races. Here, too, it is apparent that there is a general correspondence between the IQs and the coldest winter monthly temperatures and brain sizes. But once again, there are anomalies. First, the Arctic peoples inhabit the coldest zone and have the largest brain size, but their IQ is only 91. Second, the Bushmen have the second smallest brain size but the lowest IQ, while the Australian Aborigines have the smallest brain size but a slightly higher IQ than the Bushmen. Apart from these anomalies, there is a perfect correspondence between race differences in brain size and IQ. To explain these anomalies, we have to consider the genetical principles involved in the evolution of the race differences in intelligence. This question is taken up in Section 8, Table 16.2. Race differences in winter temperatures and brain size. A related approach to the issue of the relation between winter temperature and intelligence throughout the world has been taken by Templer and Arakawa. They examined the relation between average IQ in 129 nations and lowest winter temperatures. The correlation was 0.68, showing that the lower the winter temperature, the higher the national IQ. They also examined the relation between average national IQ and skin color and found a correlation of 0.92. The lighter the skin color, the higher the IQ. 5. Brain size and intelligence in humans. Thus, there is a broad correspondence across races between severity of winter temperatures, brain size, and intelligence. The explanation for this is that the colder the winters in the climatic zones occupied by the races,
the greater the selection pressure for larger brains to accommodate the higher intelligence required to deal with the problems of survival during severe winters. This theory presupposes that brain size is positively related with intelligence in humans. There has been a marked reluctance among a number of anthropologists and psychologists to concede the existence of this association. For instance, Riedel, Kleekamp, Harper, and Kretschmann reported a study showing that the average brain weight of Australian Aborigines was substantially lower than that of Europeans. But it has to be emphasized that this difference in cortical volume cannot be interpreted as evidence of intellectual abilities or intelligence in Aborigines. No study has ever shown a correlation between neuroanatomical data and intelligence in man. Bradshaw writes of the vexed issue in our species of a possible relationship between brain size and intellectual capacity. Beals, Smith, and Dodd write no convincing case for such associations has ever been presented for the association between brain size and intelligence. Similarly, there is even more evidence accumulating against a direct relationship between cranial capacity and intellectual capacity. There is really no evidence to show that brain size is positively correlated with intelligence. There is no evidence that larger brains are, in any way, better than smaller brains, and there has never been a scientifically acceptable study that supports the hypothesis that intelligence is dependent on brain size. Contrary to these assertions, the positive association between brain size and intelligence in humans has been shown in numerous studies carried out from the first decade of the 20th century. The research has been reviewed by Vernon, Wickett, Bazana, and Stelmach, who report 54 studies that used an external measure of head size. Every single one of the studies showed a positive relationship and the overall correlation was 0.18. They also report 11 studies of normal populations that measured brain size by CT and MRI, which give a more accurate measure of brain size, and for which there was an overall correlation of 0.40. A further study published subsequent to this review found a correlation for 40 subjects between brain size measured by MRI and intelligence of 0.44. Vernon et al. Conclude that the most reasonable interpretation of the correlation is that brain size is a determinant of intelligence. Larger brains have more neurons, and this gives them greater processing capacity. It is not only among humans that brain size is correlated with intelligence. The same association has been found among rats in a study by Anderson, in which rats' ability to learn their way through mazes was positively correlated with their brain weight. The correlation of 0.40, obtained by Vernon et al. Between brain size and IQ, should be corrected for measurement error of the intelligence tests. Correction for measurement error is obtained by dividing the correlation by the square root of the product of the reliability coefficients of the two measures from which the correlation coefficient is computed. The reliability of intelligence tests is typically around 0.90. The reliability of the brain size measures is not known, but it is assumed to be perfect. Correction of the correlation of 0.40 between brain size and IQ for the imperfect reliability of the intelligence tests gives a true correlation coefficient of 0.44. 6. Contribution of race differences in brain size to differences in intelligence. We now consider the extent to which race differences in brain size can explain the differences in intelligence. To do this, we have to calculate the race differences in brain size in standard deviation units and multiply the ds by the correlation between brain size and intelligence. This gives the IQ differences of the races attributable to the brain size differences. These calculations require means and standard deviations of brain size for the races. The standard deviations are only available for Europeans, Africans, Native Americans, South Asians, and East Asians, and are given by Beals, Smith, and Dodd. So these are the only races for which the calculations can be made. The results are summarized in Table 16.3. Column 1 gives the two races being compared. Column 2 gives the differences in brain size between the two races expressed as D-scores calculated from the figures given in Table 16.1. Column 3 gives the IQ difference between the two races expressed as D-scores predicted from the brain size difference, obtained as the product of the D-scores given in Column 2, multiplied by 0.44. Column 4 gives the racial IQ differences predicted from the brain size differences. Column 5 gives the actual IQ of the race in comparison with 99% for Europeans. Row 1 gives these figures for the European-African comparison. The difference in brain size predicts that Africans would have an IQ of 91. Their actual IQ is 67, so the brain size difference predicts 28% of the IQ difference. In Chapter 4, the genotypic IQ of Africans was calculated as 80%, so the brain size difference explains about half the genotypic IQ difference. The other half must be attributed to differences in neurophysiological processes. Row 2 gives the figures for the European Native American comparison. The difference in brain size predicts that Native Americans would have an IQ of 97. Their actual IQ is 86, so the brain size difference predicts about a fifth of the IQ difference. Row 3 gives the figures for the European, South Asian, and North African comparison. The difference in brain size predicts that South Asians and North Africans would have an IQ of 96. Their actual IQ is 84, so the brain size difference predicts a quarter of the IQ difference. Row 4 gives the figures for the European East Asian comparison. The difference in brain size predicts that East Asians would have an IQ of 109. Their actual IQ is 105, so this time the brain size difference overpredicts the IQ difference by 4 IQ points. There are two likely explanations for this. The first is that East Asians suffer environmental disadvantages that prevent their genotypic IQ being realized. If this is so, the East Asian IQ will rise to around 109 when their environmental conditions improve to the level of Europeans. The second is that the large East Asian brain serves cognitive abilities not fully represented in intelligence tests. The most likely of these 1/2 the visualization abilities. Although the contribution of race differences in brain size to race differences in IQs can only be calculated for the racial comparisons given in Table 16.3, the results showing that race differences in brain size explain about a quarter of the differences in intelligence can probably be reasonably extended to all race differences. The remainder of the differences are attributable to environmental inequalities and differences in neurophysiological processes. Table 16.3, Race differences in IQs predicted from differences in brain size. 7. Sex differences in intelligence and brain size. A problem that has sometimes been raised in connection with the existence of race differences in brain size and intelligence is that women have significantly smaller brains than men, and yet it has been virtually universally asserted that there is no difference in intelligence between men and women. For instance, women's brains are 10% smaller than men's, but their IQ is on average the same. Since women with their smaller average brain size are just as intelligent as men, it appears to follow that brain size has no effect on intelligence. This is the conclusion drawn by Gould, who writes that it disproves the myth that group differences in brain size bear any relationship to intelligence. The smaller average brain size of women has been shown by Ankney and Rushton. Ankney calculated that the average male brain, adjusted for larger body size, is 100 grams heavier than that of women. Rushton calculated from another data set of 6,325 military personnel that the average male brain, adjusted for larger body size, is 1,442 cc, and the average female brain is 1,332 cc, a male advantage of 110 ccs. 1 cc of brain tissue weighs approximately 1 gram, so the Ankney and Rushton results are closely similar. Thus, we have the paradox that brain size is positively related to intelligence, that men have larger average brain size than women, and yet men and women have the same intelligence. I have presented the resolution of this paradox in Lin and Irving. It is that up to the age of 15 years, males and females have approximately the same intelligence, except for a small male advantage on the visualization abilities. But from the age of 16 years, males begin to show greater intelligence, reaching an advantage of from 3 to 6 IQ points in adults. Intelligence can be defined in 4 ways, and in 3 of these, the evidence for higher average IQs in men is quite clear. First, intelligence can be defined as the full-scale IQ of the Wechsler tests, which provide the average of all major abilities including verbal comprehension, verbal and nonverbal reasoning, visualization, perceptual ability, immediate memory, and perceptual speed. On 5 standardization samples of the tests for adults, the average male advantage is 3.51 Guatemalan Quetzal points. A more recent study by Colom, Garcia, Juan Espinoza, and Abad of the Spanish standardization sample of the WACE 111 reports an almost identical male advantage of 3.6 IQ points. The average male advantage of 3.5 IQ points among adults on the full-scale IC of Wechsler tests obtained from the 6 standardization samples underestimates the true male advantage for 2 reasons. First, the tests are biased in favor of females because verbal abilities, on which the male advantage is relatively small, are overrepresented with 6 subtests, while visualization abilities, on which the male advantage is larger, are underrepresented with only 2 subtests. Second, in the construction of the tests, a number of items favoring either males or females were eliminated, which would tend to produce equal IQs for males and females. A second approach to the issue is to define intelligence as nonverbal reasoning ability as measured by the progressive matrices. This is the definition adopted by McIntosh, and the test is widely regarded as one of the best tests of G. It has been asserted by Court, McIntosh, and Jensen that there is no difference in the mean scores obtained by males and females on the progressive matrices, and therefore that there is no difference between males and females in reasoning or in G. Contrary to these assertions, a meta-analysis of all 14 known studies of adults has shown that men invariably obtain a higher mean IQ than women on the progressive matrices by an average of 5 IQ points. A third approach to the problem is to define intelligence as the average of nonverbal reasoning, verbal comprehension, and visualization abilities, as proposed by Gustafsson. Data assembled from 13 countries show that on this definition the mean IQ of adult men exceeds that of women by 4.9 IQ points. The most satisfactory definitions of intelligence are to follow McIntosh and define it as nonverbal reasoning ability, or to define it as the average IQ of nonverbal reasoning, verbal comprehension, and visualization abilities. The 2 definitions yield similar results. On the first, there is an adult male advantage of 5 IQ points, while on the second, there is an adult male advantage of 4.9 IQ points. A fourth approach is to define intelligence as the G extracted from a battery of tests containing all or most of the major primary abilities. This definition has been adopted by Jensen. He presents the results of 5 studies. The results were that males obtained higher IQs of 5.49 IQ points on the ASVAB, 0.18 on the American Standardization Sample of the WACE, and 2.8% on the American Standardization Sample of the WISC-R, while females obtained higher IQs of 0.03 IQ points on the BACE and 7.9% 1 IQ points on the GATE-B. From these inconsistent results, he concludes that sex difference in psychometric G is either totally non-existent or is of uncertain direction and of inconsequential magnitude. Jensen's conclusion does not resolve the problem that males have larger average brain size than females, yet according to this analysis have the same average IQ. Jensen suggests that the resolution of this problem may be that males and females have the same number of neurons, but the female ones are more densely packed into a smaller brain. It is improbable on general biological grounds that a sex difference of this kind would have evolved, and it has been disconfirmed by Packenberg and Gunderson, who found that men have more neurons than women, but there is no difference in neuronal density between male and female brains. There are 2 problems with Jensen's conclusion that there is no sex difference in intelligence. First, he does not acknowledge the evidence that I set out in Lin, and that has been confirmed by Colom and Lin and Irving that the male advantage is not present, or is minimal up to the age of 16 years, so the results of the WISC-R on 5 to 16-year-olds and of the British Ability Scales on 14 to 17-year-olds need to be set aside. Second, the inconsistency between the 5.49 IQ male advantage on the ASVAB and the 7.91 female advantage on the GATE-B is so great that something must be wrong with the method. The problem is that the nature of the G extracted from a battery of tests is affected by the kind of tests in the battery. A predominantly verbal test like the Wechsler's yields a verbal G on which on the adult male advantage is quite small. The GATB on which there is a female advantage 7.9 IQ points on G contains a number of perceptual and psychomotor tests and hence yields a perceptual psychomotor G females perform better than males on these tests, and so, as Nyborg correctly observes, they have a higher G. If these tests are removed and the tests of verbal, numerical, and spatial abilities are analyzed, the female advantage disappears, as Jensen shows. More recent studies using this method show further inconsistent results. Colom, Juan Espinoza, Abad, and Garcia, analyzing a large Spanish sample, and Colom, Garcia, Juan Espinoza, and Abad, analyzing the Spanish standardization sample of the WAIS 111 by the same method, found that the sex differences in G were negligible. However, using the same method, Nyborg has obtained a male advantage among adults of 5.5 IQ points, and Colom and Lin, in an analysis of the Spanish standardization sample of the DAT, obtain a male advantage among 18-year-olds of 4.3 IQ points. It is evident that the sex difference obtained by this method is highly variable. The reason for this is that different tests produce different Gs. The higher male IQ can be ascribed to the larger male brain. 3 studies have shown that the average male brain size exceeds that of the female brain, corrected for body size. Ankney found a larger brain size measured by weight of approximately 100 grams. Rushton found a difference in volume of 110 cc's, and Tan et al. Found a difference among college students in Turkey of 91 ccs. Ankney expresses the male-female difference as 0.78 d. The correlation of brain size with intelligence is 0.44, hence the predicted male advantage in intelligence arising from a larger average brain size is 0.778, multiplied by 0.44, giving a male advantage of equal to 5.1 IQ points. This should be considered the same within the range of error arising from the use of different tests, measurement error, and different definitions of intelligence, as the actual male advantages of 4.9 IQ points estimated in Lin, 5 IQ points estimated in Lin and Irving, 5.49 IQ points on the ASVAB, 5.55 IQ points found in a Danish sample by Nyborg, and 4.3 IQ points found by Colom and Lin in a Spanish sample. The average of the 4 estimates is 5 IQ points and should be adopted as the best estimate of the male advantage among adults. This advantage is wholly explicable by the larger male brain, as would be expected because males and females experience the same environment and therefore environmental factors cannot account for the male-female difference. The explanation in evolutionary terms for the greater average intelligence of men is probably that in most social species, males compete with one another to obtain female mates, and in the evolution of the hominid's intelligence came to play a significant role in success in this competition. Females do not compete for male mates to anything like the same extent. This would have exerted stronger selection pressure for enhanced intelligence in men than in women. 8. Genetical processes in TBE evolution of race differences in IQ. 2 genetical processes must be assumed to explain the evolution of race differences in intelligence. The first of these is that differences in the frequencies of the alleles for high and low intelligence have evolved between races, such that the alleles for high intelligence are more common in the races with the higher IQs and less common in the races with the lower IQs. The early humans that migrated out of Africa and spread throughout the world would have carried all the alleles for high and low intelligence with them. But those who colonized Asia and Europe were exposed to the cognitively demanding problems of survival during cold winters. Many of those carrying the alleles for low intelligence would have been unable to survive during the cold winters, and the less intelligent individuals and tribes would have died out, leaving as survivors the more intelligent. This process would have reduced and possibly eliminated the alleles for low intelligence, leaving a higher proportion of the alleles for high intelligence. The more severe the winter temperatures, the greater the selection pressure for the elimination of low IQ individuals carrying low IQ alleles. This process explains the broad association between coldest winter temperatures and IQs and brain size shown in Table 16.2. A parallel genetical process must have been involved in the evolution of race differences in skin color. The first humans who evolved in tropical equatorial Africa must have had black or very dark skins, as these peoples do today, because of the adaptive advantage of dark skin in strong sunlight. When some of these early peoples migrated into North Africa, Asia, and Europe, alleles for paler skins must have appeared as mutations. Individuals with these mutations would have had a selective advantage because they could synthesize vitamin D from sunlight, while at the same time they did not suffer the disadvantage of contracting skin cancer from the excessively strong sunlight of the tropics. Hence, individuals with paler skins left more surviving offspring, with the result that the alleles for paler skin spread through the population and eventually replaced the alleles for dark skin. This process produced the same broad gradient for skin color as evolved for intelligence, with the Arctic peoples, East Asians and Europeans having evolved the palest skins, the South Asians and North Africans, Native Americans, Southeast Asians and Pacific Islanders having evolved somewhat paler skins, while the Africans, Bushmen, and Australian Aborigines exposed to the strongest sunlight retained the darkest skins. A second genetical process has been proposed by Miller, in which several new alleles for high intelligence appeared as mutations in some races but did not appear in others, and these were never transmitted to some other races. This assumption is necessary to explain some of the anomalies in the general relationship between severe winters and the race differences in intelligence. The general principles are that new mutant alleles for high intelligence would be most likely to appear in large populations and in populations that are subjected to stress. New mutant alleles for high intelligence would be most likely to appear in large populations because a mutation is a chance genetic event, and hence is more likely to occur in races with large populations. In addition, populations subjected to stress, including extreme temperatures, also experience more mutations. The effect of these two principles is that mutations for higher intelligence would have been more likely to occur, and can be assumed to have occurred more frequently in the South Asians, who had large populations and were subjected to cold stress, and particularly in the East Asians and Europeans who had large populations and were subjected to extreme cold stress, than in the Africans who had a large population but were not subjected to extreme cold stress, and in the Australian Aborigines and Bushmen who had small populations and were not subjected to extreme cold stress. The Arctic peoples were subject to extreme cold stress, but comprised very small populations, so they would be unlikely to have had mutations for higher intelligence. It may also be that directed mutation also operated to produce new mutant alleles for high intelligence in the South Asians, and particularly in the East Asians and Europeans. The concept of directed mutation is that a mutation is more likely to occur if it is advantageous to the organism. The theory was first proposed by Cairns, Overbaugh, and Miller, and has been supported by a number of biologists. Higher intelligence would have been more advantageous for the South Asians and particularly for the Fast Asians and Europeans than for the Africans. Once a new mutant allele for higher intelligence had appeared, it would confer a selection advantage and would have spread throughout the group of around 50 to 80 individuals in which people lived during the hunter-gatherer stage of human evolution. It would then have spread fairly rapidly to adjacent groups because hunter peoples typically have alliances with neighboring groups with which they exchange mating partners, and it is reasonable to assume that this custom was present for many thousands of years during the evolution of the races. These alliances of groups are known as demes, and a new mutant allele for higher intelligence, and which conferred a selection advantage, would have spread fairly rapidly through a deme. From time to time, matings would take place between demes, and by this means new mutant alleles for higher intelligence would spread from one deme to another, and eventually throughout an entire race. It has been estimated by Rouhani, using reasonable assumptions of a selection coefficient of 0.01 and a 5% migration per generation between hunter-gatherer demes of around 500 individuals, that advantageous alleles would spread at a rate of 0.8 miles a generation. Thus, in 25,000 years, consisting of approximately 1,000 generations, an advantageous allele would be transmitted about 800 miles. Hence, an advantageous allele occurring as a mutant in the region of, say, Beijing, 25,000 years ago, would not yet have spread outside China, and would take another 50,000 years or so to reach the Arctic peoples of far Northeast Asia. This model does not, however, take account of the geographical barriers that have generally been present between the races, such as the Gobi Desert between East Asians and Europeans, and the Sahara between sub-Saharan Africans and North Africans, which have largely prevented interbreeding between the demes of different races, and hence the transmission of new mutant alleles for higher intelligence from one race to another.